So our project, I'm Joan. Helen, who has done most of the heavy lifting for this project, is going to be the second part of our talk. Ken Jones, who's in the back here, does our basic informatics, and Trevor does all the annoying administrative things that somebody has to do for this project. Okay. So, yeah. Okay. So the problem is the developmental programs that take a mouse face from a series of bumps to something that looks like a face. And so what we see here is a set of static images, but really development is a very dynamic process. And a lot of what we try and do is reconstruct the dynamics from our static images. And whether that would be from anatomical images or from sort of molecular biology kinds of static snapshots, it's a similar kind of reconstruction problem. And so we're taking a stab at that dynamics in the following way. First of all, we're collecting transcriptome data from different stages at fairly high resolution in both time and space and tissue layer. We're looking at the larger transcripts, link RNAs, coding RNAs. We're looking at the smaller transcripts, the microRNAs that we think about, but also smaller regulatory RNAs like snRNAs and SNO-RNAs. In addition, because of the issue of the dynamics, there are lots of regulatory features that are embedded in these RNAs, particularly in the UTRs, differential uses of promoters, differential splicing, that will affect the regulatory potential and the dynamics of these messages that are encoded. And then finally, we actually want to look at the translation dynamics itself. And so this is really quite important. There are estimates that maybe only half of the abundance of what we see in terms of proteins is actually explained in level of abundance of the mRNAs, and most of that is because of differences in translational dynamics. So this is the kind of data set that we're trying to assemble to see the dynamic from our static snapshots, the dynamics of the developmental programs. And the approach we're taking, or I should say what Helen has done in most heroic work, is 3 different ages, bits of the mouse face, 3 different prominences, separating them into ectoderm and mesenchyme, and then out of that to isolate either the RNA or, in the near future, the ribosome-associated messages, and then subject these to the various analyses to collect the data. And so in terms of our first aim, the static transcriptome, that's really pretty well complete. In terms of the larger RNAs that we're looking at by RNASeq, we've got 20 different samples, triplicates of each, so 60 samples. It's quite remarkable depth, 100 million reads per sample, and the data seems to be quite high and reproducible, that the replicates cluster together very nicely, that they separate along principal components by tissue layer. Ectoderm versus mesenchyme. Yes, here we go. And by age, and also by prominence. We also have a data set of the smaller RNAs, and we thought long and hard about the best way to do this, and thought that we would give a stab at actually measuring these with microarray technology instead of RNA-seq technology. There's all kinds of issues with quantitation of small RNAs. And so we designed a custom microarray that has all the known microRNAs, microRNA precursors, as well as a series of these other regulatory RNAs, the SNO-RNAs that are involved in covalent modifications of RNAs. The snRNAs are involved with the spliceosome, as well as tRNAs and ribosomal RNAs. So we designed this array. We ran our 60 different samples across it and came up with several hundred significantly differentially expressed small RNAs, which Again, in terms of the quality of the data and the principal components, they different samples group very nicely by age and also by prominence, and also not shown here by tissue layer. In terms of what we see in the differential expression, I'm going to start with the microRNAs because that's what we think we know what to expect. What I will say is that we got a little bit of a surprise here. If we, in our large RNA data, and what Yang also talked about, things separate out very nicely by tissue layer, by or by age. But in the microRNAs, it's a much more heterogeneous and salt and pepper kind of a pattern. So we see things that separate out here in the by age. So these are groups of microRNAs that are expressed early but not late in both the ectoderm and the mesenchyme. We see things that are expressed primarily in the mesenchyme but not the ectoderm. We see things that are expressed primarily in the ectoderm but not the mesenchyme. We see odds and ends of things. This is a set that is expressed really only in the neuronal epithelium that's going to be making the olfactory sensory organ, but not much else. And so we see a whole lot of variety here. If we look now beyond those microRNAs and some of the other regulatory RNAs, for instance, the SNO RNAs that are involved in covalent modifications of ribosomal RNAs, let me just say that most of these small RNAs, the SNO RNAs, the tRNAs, the ribosomal RNAs, were not differentially expressed across the data set, but we found a small subset that were in each of those classes, and I think this is really food for thought. We don't really have much to say about it at this point beyond that. We have a small set of these SNO RNAs that are really in the ectoderm increasing with time. We have another small set which is really in the mandible, but both ectoderm and mesenchyme. We have no idea what these are doing, but we would like to think that they're perhaps involved in the ribosomal diversity that might underlay some of the tissue differences. If we look at the snRNAs, so these are spliceosomal RNAs, we see some U6 RNAs that are differentially expressed in kind of odd pattern. We also see some U1 RNAs that are differentially expressed increasing with age, which again we would like to think might be involved in diversity of spliceosome activity. And then we also see a few tRNAs with quite odd expression patterns that are here and there and everywhere. So we don't really understand any of this and have much to say about it, except that we think that this is part of the story that we need to consider when we think about the dynamics of the entire genomic programs. All right. So that pretty much wraps up my quick overview of the data that we've actually generated about the static transcriptome pattern. That data, I believe, is all uploaded to the hub now and in the process of being turned into track hubs and things like this that are to be available to the public. Now what I want to do is to move on a little bit and talk more about the isoform analysis, which is really the nuts and bolts of what we're doing these days. So whereas this aim was really about data acquisition, this is really about data analysis. And I want to make the argument to you that this is really important. These different isoform features have a huge amount to do with what actually happens in our genetic programs. So just in terms of the diversity that's out there, more than half of our genes have alternative promoters or alternative polydenylation sites. But beyond that, there's pretty good estimates that we only know about, have annotated about half of the alternative promoters that are out there. So there's a lot of diversity, and there's a lot of diversity we don't know about. And I will also make the argument that those alternative promoters or alternative UTRs are really important because that's where the regulatory information resides that is going to be regulating the translational dynamics, a big piece that we just don't know about. And so analysis of the isoform features, how are we doing it? What are we doing? So we're starting with the RNASeq data and looking for either differential promoters. So this is an example of a promoter that's in the ectoderm, a promoter that's in the mesenchyme, alternatively used, differentially used. An example of a 3' end differentially used in the mesenchyme versus the ectoderm. And a couple of different flavors of alternative splicing, an exon that's skipped in the mesenchyme or alternatively used in mesenchyme and ectoderm. So these are specifically the kinds of differential usage that we're looking for. How do we do that? Well, it's not so obvious and it's not so easy. And, we blithely said when we wrote our proposal that we were just going to find the different isoforms and count them. Well, it turns out you can't get there from here. We cannot accurately reconstruct isoforms and their abundances from standard RNA-seq data. There is just too complex a problem. Beyond that, to accurately measure this stuff, you need really good gene models. And the gene models that we have right now just aren't good enough. They don't accurately reflect what's out there. And beyond that, we thought from our standard RNA-seq data, we need to be able to find the ends. And so there are solutions for all of these problems. First and foremost, since we can't do it, we're ignoring the problem. And instead, we're focusing on what we can do, which is measuring the differential exon usage and splice junction usage. In terms of the gene models, well, that's not really such a problem. We just have to actually look in our data and build good gene models from the data that we have. And finally, although the vanilla RNA-seq is not much good for finding the ends, there are very good databases of ends. And so we're using those databases and some custom scripts to actually estimate where everything is to be able to solve these problems. And so just briefly, this is kind of what our pipeline looks like. We start with all these different databases, the ensemble gene models, the transcription start site database, poly-A site database, splice DST information, and our own RNA-seq data, and put them together to come up with really improved gene models. And then with these improved gene models, we can feed them through software which is designed to either look for differential splicing or differential exon usage. So then in the end, we can start to model the dynamics of how these things affect actually the transcriptome and how it gets turned into dynamic programs. Okay. So just a little bit briefly about these new gene models, because this is the part that we're really pretty confident about. So beginning with what's in the ensemble gene models, there's about almost 50,000 genes, more than 100,000 transcripts, more than 100,000 transcript start sites, 400,000 exons. About 20% of those are not actually expressed in our data, but the remaining 80% are, more or less. But in addition to that, we found a whole series of new genes, exons, transcripts, transcription start sites, polyethylation sites. There's some blanks down here. We haven't quite finished this analysis at the 3' end. But what I want you to focus on is all this novel stuff that we've found that has come out of our RNA-seq data. And we've been able to now incorporate that into really a face-specific set of gene models, which we can then go on to use for our various analyses. And I just wanted to give you a flavor of what we found, and partly because it relates a lot to some of the other talks we've had earlier, talk a little bit about some of these novel genes that we found. Okay, so almost 5,000 of them, and threshold, they have to be found in more than one sample, at least 10 reads per sample, no overlap with exons of known genes. So that's what I'm defining as new genes. And because my collaborators are quite skeptical of everything, I had to prove to them that these really were not just garbage, mismapped sequencing reads, and various other kinds of junk. So overlap them with things, various databases that we know about. Turns out that about 1,000 of these actually are represented as ESTs already, but not ensemble annotated genes. A few of them are annotated pseudogenes. A few of them are entirely contained within repetitive elements, LTRs. And so those are probably mapping garbage. So probably we have about 20% garbage, and about 80% interesting new stuff. And what do these things look like? Most of them, these are the violin plots that show length in terms of nucleotides. So most of them are very short, less than 500 nucleotides, although the genes that are in EST clusters are much longer. Most of them have quite low gene expression level, a few less than 10 FPKM, that would be about here. Although again, the ones that are in the EST clusters tend to be significantly higher or have a subset that are higher expressed. In terms of exons, most of them are single exon, although again, the ones in the EST clusters tend to have more exons. And this is what I found really provocative. Many of these ones have ambiguous strand, which means they have lots of reads on those strands. So when I look at this set of properties, single exon, ambiguous, both strands, short, low expression, it makes me think about enhancer transcripts. And so I'm very interested in this group of novel genes, is about 2,500 of them, and thinking that maybe a lot of them are actually representing bidirectional transcripts off of enhancers. And again, just an example of one, this is one that we found just here at the 3' end of the RAB28 locus. So about 300 nucleotides in between repetitive elements, just downstream to the 3' end of RAB38. And if this were really an enhancer RNA, it should have correlated expression with a nearby gene. So this is actually the expression profile of this little transcript pile, higher in the ectoderm in the mandible. And this is the adjacent RAB38, which has a pretty similar expression profile. Two flanking genes, the CTSC and the ribosomal pseudogene, look different. So, for instance, this is a candidate enhancer for the RAB38 and the transcript files. So that's really just what I wanted to say about these novel genes. Summing up by saying that we have a set of things that are represented by ESTs that are probably mostly regulatory long non-coding RNAs. We have a set of things that are mostly small single-stranded that are probably a lot of enhancer RNAs, and we're quite interested in working with Axel's group to go through some more of this. Okay. So that wraps up really what I wanted to say, that we've developed novel gene models. And now I just want to have Helen come up and give a few words about how she's been using these novel gene models and the different software for estimating alternative usage to come up with sort of these dynamic interpretations.
Okay, thank you, Joan. Before I start, I just want to say, so as Joan mentioned, so we use the RNASeq, and then right now we're starting to do validation for the alternative splicings by qPCR and also by in situ hybridization. Like in here, this is a particular gene actually has a promoter specific ectoderm-specific promoter usage, and also has like a promoter used by both mesenchyme and ectoderm. And this is a really pretty result from our current rotation student, Nina. So as Joan had mentioned about the alternative splicing, so question is this really important for facial development? Yes, it is. So ESRB1 is epithelial-specific splicing regulator. This gene is involved in EMT and also in cancer. So our collaborator, Ross Costin, locked up this gene. So interestingly, so the lockout actually has 4 penetrance of orofacial collecting, which means alternative splicing is really important for facial development. So then the question is, what is this alternative splicing program in facial development? I would tell you about this in my first part of the talk, and then so what are the regulators for the splicing program, and I will tell you about this in the second part of my talk. So here we take differentially expressed gene and also alternatively spliced gene for one comparison, like 11.5 maxilla ectoderm versus mesenchyme with this different cutoffs. So I want you to pay attention to this one is the PSI, delta PSI. So it's the differences between the percentage splicing is larger than 10% for here. So from this figures, clearly there are a lot of genes are differentially expressed in facial development between the two tissue layers. And also there are a lot of genes alternatively spliced. So, but when we compare these two lists, there are only really small portion of genes are overlap. And so these genes are only regulated by alternative splicing. So which means alternative splicing is a major regulator in gene for gene expression in face. Here, actually, so the blue shows the alternative splicing differences between layers. Red show the alternative splicing differences between age, and then green shows the differences between prominences. So from here, clearly, first we can see, so in all three categories, the differences between layer and age actually dominates the changes. And there are a little bit change between prominences. And second thing here is like for first exon gene body or the life exon, the three categories, actually the gene body dominates three. It's the major alternative splicing in face. So since limited for time, so I will just focus on the alternative splicing for gene body. There are 5 categories, different types of gene body splicing even. I don't have time to go through every one here, so clearly from the table here, the number here, so the skipping exon is the major splicing events in facial development. To get an idea for at a systematic view, so after we've done all the comparisons between layer, between time points, and also between prominences, so I combined like all the PSI from each sample and I made this heat map. So red means higher inclusion, blue means lower inclusion. And so this is the samples from ectoderm, and this is samples from mesenchyme, and that's time difference, and then that's 3 different prominences. So clearly we can see here 3 categories came out, stands up. So one is the age difference cluster, and then the second one is the ectoderm high inclusion cluster. The third one is the mesenchyme higher included cluster. To get an idea about what's going on for this clusters, so we did geotome analysis from David. So here, clear in cluster 3, GT-based activated activities, these categories actually are highly enriched in cluster 3. So this actually gives us an idea for what's going on for the alternative splicing in facial development mechanistically. So my second part is about, so what's the regulators of the alternative splicing in face? To get an idea for that, so we started with taking a look at the expression levels of all the non-iron binding proteins in face. So here we can see a cluster actually increases over time, and also cluster increases highly enriched in ectoderm, and cluster mesenchyme express, and this will actually decrease over time. Let's take a look at some of the clusters in detail. So here, just a heat map show the highly enriched ectoderm iron binding proteins. So clearly, ESRP1 show up here. And also besides ESRP1, we see a lot of other iron binding proteins highly enriched in ectoderm. And these are the tools I will come back later. And for the mesenchyme highly expressed iron binding proteins, it's more diverse. So here is a late program, and then this is an early program, and this is more generically expressing mesenchyme. So just show 2 examples. I will come back later. So here I actually take the alternative splicing events cross reference with the iron binding motifs bound by iron binding proteins, and then I put like the also combined with the expression level of these genes. So as shown here, ESRP1 is highly expressed in ectoderm in red, and then RBMS3 highly expressed in mesenchyme in blue. So I mapped like the motifs to these regions. So here is typically skipped exon. That's the upstream exon or downstream exon, which is always included. So here clearly we can see in all these regions, actually there's a lot of enrichment for different kind of iron binding proteins. If you look at this at a more detailed view, so let's take ESRP1 as an example. This is a motif map of ESRP1. So the peaks here shows the enrichment of the ESRP1 binding site. So what does the color mean? I will show you like in this diagram. So in ectoderm, and so the ESRP1 binding site is here, and then if ESRP1 actually binds to this intron, like upstream intron of this exon, so this exon tends to be skipped. And when actually ESRP1 binds to the downstream of this exon, so this exon tends to be included. However, in the mesenchyme, so there's no ESRP1 here, so this exon is included. And then when there's binding site in the downside, there's no ESRP1. So and also so it goes to the other way, requires ESRP1 to include it, but there's no ESRP1, so this one is skipped. So that's why, like in the previous picture, you see a red peak like in the right side of the exon, and then a blue peak in the left side of the exon. What about the other iron binding proteins? Are they similar or they are acted differently? So let's look at another example, RBMS3, which is highly expressed in mesenchyme. So we can see here actually it's quite different from the ESRP1. So in the downstream intron here is a blue peak, and then like ESRP1 has a red peak, which means actually RBMS3 actually, if it binds to the downstream intron, actually increase the inclusion of this exon. So I think that's that's what we are now. There's a lot of data to sift through and also to understand. So hopefully I give you guys an idea for where we are now and also where we are going. With that, I would like to conclude with this. So for everyone, we have a generate or comprehensive data set for expression for small RNAs. And for AIM2, I just Joan and I just told you about like the alternative splicing is a major regulator for differential for expression in facial as well, and also in combined with other genomic data, so we can build a better gene regulative network, and also with the alternative splicing program and the regulatory program. So we should be able to build alternative splicing program for base. And then for AIM3, so we will characterize the translation efficiency across facial development, and also integrate this data with the data I have mentioned before to build a translational regulatory work for facial development. So with that, we will take questions.
Thank you, Joan. This guy questions.
Yeah, so with regards to the new gene models, have you looked at whether these new gene models are positionally biased towards known genes or known enhancers? And a related question to that, in terms of their total coverage, what is their?
Okay, so with regards to the second question, the coverage, I couldn't say exactly. There's certainly a large number of bona fide transcripts that genes we know and love that have the same kind of coverage. So it's certainly in the ballpark. But what was your first question again?
So whether these novel genes are they positionally biased?
I think I actually took that slide out. So the question is, where are these novel genes? They're fairly evenly distributed. Some of them are close to known genes, promoter proximal. Some of them are at the 3' end, some of them are introns, and some of them are more than 10 kb away from known genes. They're just all over the place. That answers your question.
They're according to potential, are they coding?
I haven't dug very deeply into the coding potential, so I just don't know.
I'm from China. I have a question from you. So you showed before of validation of the opponent splicing with precision. So I want to know what length of the probe you use and how do you do that?
So actually travel design the probe. So because the issue is for the alternative probe model usage, so we only have really limited lengths we can design probes. I think like the probe is around 200 the base pairs. And then we design like a probe specific accident, like only express the accident, the promoter only use the accident, like one probe for that. And then another probe actually is just like detect like all the other promoter express both in accident and medical. Because they use different promoters, but then the length is one, and then we divide programs on it. So we have top one, and then that one was the one worked with.
Yeah, and just one more. You're a little over time, but.
So I guess I would ask, not getting too metaphysical, how do you define a gene? In the old days, we would have called most of what you keep calling genes transcription units. And I'm just curious what you mean.
Yeah, that is metaphysical. So I suppose when I'm calling these things genes, what I should call them is piles of read. And that's really all we know right now. And you know, our classically we think of a gene that has something with transcription start and transcription stop, and it has meaning. And I think a lot of my read piles that are probably associated with cancers don't fit that definition. So, but the way I was very specifically defining them is read piles that don't overlap known exons. So for the purposes of today, that's how I was defining.